And so today we have on with us actually our Northeast Rural Forester, EJ Jameson, who thank you for hitting record, EJ. Good job, team effort. EJ is going to be helping moderate chat since I'm here with Sherry today. So we're very glad to have her on. For those of you that haven't joined us before in book club, we are recording. So this will be posted on YouTube for anybody that cannot make the live session. We try to do a couple book clubs a year. So if you have any suggestions about book club or questions, comments, concerns, please feel free to send those my way, as we always want to improve that process. But today we're going to start with Sherry giving a presentation. And then once she's done, I would love for you all to ask questions. We do have a couple photos of mushrooms for her to identify, but if you all have questions, if you want to comment on the book or just ask questions about mushrooms in general, that's what this time is here for. So without further ado, I'll go ahead and start sharing my screen, and then we will have Sherry get started.
Okay, there it is.
Perfect. And I'll move this out of the way so you can see yourself.
Oh.
There you go.
Okay. Well, I'm Sherry Kay, and this presentation is about the book, A New Guide to Kansas Mushrooms, which I put together with a great deal of help from Ben Sykes, an associate professor and scientist of microbial ecology at KU. And if he shows up, because he said he might show up to this, he's very welcome to say a few words whenever. And Caleb Morse, the collections manager for the Division of Botany at KU's Biodiversity Institute. And again, Caleb, chime in if you show up here. I invite you both to do that. So this new guide began as a second edition of a guide to Kansas mushrooms, which was produced by my husband, Richard Skip Kay, a couple of friends, Bruce Horn and Dean Abel, and myself about 30 years ago. And it was published by the University Press of Kansas. So I'm going to give you a little history about how these books came into being. I grew up in the middle of Kansas, born in Kingman 77 years ago, and essentially could say I've hunted mushrooms for over 70 years, including walks with my mother when I was 4 or 5 years old. The earliest Kansas mushroom guide I used was Ansel Stubbs' book, produced with the help of Dr. Robert Lichtwart, the mycologist at KU for most of the last half of last century and active into this one. I used this guide to help supplement my food supply when I went to graduate school here at KU over 50 years ago. I met Skip Kay, professor of medieval history, through mutual friends. He enjoyed the outdoors, but at the time we married, he was very sedentary and entirely absorbed by his scholarship. He needed to get outdoors again, so I introduced him to the joys of the mushroom foray. We included a number of friends, including Dean Abel, Bruce Horn, in our mushroom forays. And I took Bob Lichtwart's introduction to mushrooms. Soon Skip applied his considerable organizational skills. Do we have a?
I think we have somebody who needs to mute. There we go. Okay.
Okay, we got it. Okay, so Skip applied his organizational skills to make the Kaw Valley Mycological Society. After hours of hunting mushrooms, we spent hours trying to identify them using books and microscopes. At first, most of our catch was mysterious, but slowly we became more knowledgeable. Skip was a superlative Latinist, and we tried to identify using the Latin names. Affiliated with the North American Mycological Association and greatly benefited from their national forays as well as the materials they sent to us. And they're still going. So if you guys are interested, I know they have an online presence. The lure of scholarship got to Skip, and he became editor of our newsletter, as well as compiling a checklist of Kansas mushrooms, a heavily annotated book listing all the mushrooms found in Kansas to that time. The University Press of Kansas learned of our activities and asked us to do the first guide to Kansas mushrooms, and now we have a new guide to Kansas mushrooms. This project began about 8 years ago. It took about 6 years to get it out into the world. And that was when the press approached us to do a new edition of the original guide. Skip was very ill and declined. Bruce and Dean were consulted, but they were also unwilling, so I thought I'd have a go. Skip did help me, and I remember sitting in the ER for 4 hours with him planning this book. He died in 2018, and he was the best life partner I could have ever had. There's been a general increased interest in mushrooms, especially after that movie, Fantastic Fungi. There are also 2 other very good reasons to update the old book. First, at the time of writing the original book, we'd only found about 725 different mushrooms. Now the count is well over 1,200. Therefore, we've added 50 more pictured species to the new book, and many more are discussed as well as included in the keys. I would say that there are actually more like 5,000 different mushrooms in Kansas, because every time I go out looking, I find stuff I don't know what it is and can't identify it. The second reason is that the gene sequencing revolution has improved our knowledge of the relationships among mushrooms, making necessary changes to individual mushroom names as well as the names of the higher groupings. This is because the scientific names of a mushroom should reflect in some way its place in the whole fungal kingdom. The result has been that about 25% of the names in the first guide are out of date. It's actually more than that now. At the end of the new guide are some amazing cladograms which illustrate with text and diagram the relationships between the mushrooms in this book. Ben Sykes is responsible for this extraordinary feat. Now, you don't have to understand this or deal with the science at all in order to use this book to identify mushrooms. Caleb has provided excellent keys using observable with the naked eye characteristics that will lead you to identification, and we have given common names if there were common names. We did not make up common names, however, so many of the species, in fact, most of them, simply only have the scientific names. We have all 3 read and revised every word of this book. It seems like endlessly, but now it's done. And I think it's usable for anyone who is interested in mushrooms. So are there any questions at this point? Well, we will go on if that's all right. And I will show some energy. I want to say one thing. I hope everybody understands that mushrooms are the fruit of a not easily seen organism that's underground or in wood or something like that. It's pretty sensitive to UV and it has to protect itself. So it's just the fruit. So I will show you some images now. And the first one is Skip K with the most marvelous collection of morels we've ever found. They grow around cedars right around the drip line. I don't know. They really could have a relationship, but cedars are pretty hostile, so it's hard to believe. So the next one. And this is Bob Lickford and I looking at mushrooms, and Dog Charlie's probably looking at the goodies. And next is Dean Abel with a microscope, and he was a professional microscopist. And next is Bruce Horn, dwarfed by mushroom rock in western Kansas. I couldn't get a better one. He helped us by lending some much needed mushroom slides for the new book. And next. And this is Hank Guerisco, specializing in spiders, but he's a great nature photographer and he loaned us some beautiful images. And next. And this is Carla Wick with mushrooms I can't identify. She is another fabulous nature photographer who has allowed us to use a number of her amazing images. Okay, so now I'm going to show some mushroom images. And okay, so mushrooms are the fruiting body. They're also called macrofungi, as they are mushrooms that can be seen with the naked eye, and fungi, period. And I've grouped them here as they are grouped in the new guide according to phylum they belong to, or phyla, which is plural. And these are the major groupings in this kingdom. And the 2 here represent most of the macrofungi. The Basidiomycota have guild mushrooms like the grocery store types. The Ascomycota have the most sought after Kansas mushroom, the morel, but also have a multitude of others. The big difference between them is microscopic. The basidiomers grow on the tips of clubs-like structures, and the ascos are contained in these sacs. This is the spores. We're going to begin with the basidiomycetes, and that is a basidiomycete. I've got a lot of different images than I have in the book of the same mushroom, and then some images of mushrooms that aren't in the book. So I thought you'd just be a little different. This is Rhodotus palmatus by Carla. This showy mushroom seems to be found in Kansas more often than other states, and we used to ship them over to people who were interested. Mushrooms often have structures observable that produce the spores. It's often on the underside, closer to the ground. And in this case, they are gills, which are in the grocery store mushrooms too. And they are sort of blade-like projections from the underside of the cap, moving from the stem to the edge of the cap. Okay, so we'll go one more. And there are a lot of bright colored mushrooms, and among the russulas, there are many different red ones, hard to distinguish from each other. So I can't even give you a name beyond russula here. Some of these red russulas are really peppery to taste, as are some other mushrooms. And the next. Here's a green russula, Russula vrescens. It looks moldy, but is a great edible. Still, you should be careful because there are some poisonous mushrooms. Russulas. An important characteristic of russulas is their brittle stalk that breaks like a soft piece of chalk. And the next. There are a lot of purple mushrooms, especially in the genus Cortinarius. The big distinguishing character for Cortinarius is the cortina. And that is that spider web-like cover rusted down from the gills. The other is younger, and the spores are not yet ripe. The cortina can have its own color, later obscured by the spores. And of course, that's one of the ways you might identify which particular Cortinarius it is. This cortina is very fragile, and so you need to look very young little spiderweb fragments on the stalk, but mostly you need to just look for a young specimen. Okay, so next. And that is Chlorophyllum molybditis. It's a common lawn mushroom and also the most common poisoner. Not deadly, but if eaten, you should go to the ER as there is danger of dehydration. I understand it tastes great going down, but I'm not sure it's worth it. The cap is highly variable and appear in tan or pure white, and the scales are not always noticeable. The spore color is a big way to identify this mushroom. It's the only mushroom I know of with dusty green spores. The gills on the underside are not shown here, and they will be colored green by mature spores, but the gills seem to stay white for a long time, and in dry weather, especially, I've seen the gills look brown. So we go to the next one, and this is Amanita populophylla. It has a special symbiotic relationship with cottonwoods. Actually, KVMS, that is Kaw Valley Mycological Society member Elizabeth Moses, initially found this over by Manhattan. And with the world-famous amanitologist Rod Tullis named this to science. So this is the original from Kansas, populophylla. I think it should be our state mushroom. So anyway, it has a special symbiotic relationship, that mycorrhizal association with cottonwoods. You really only find it with cottonwoods. So the mushroom organism, looking cottony or web-like, minutely covers the root tips of trees and some other green plants, and then exchanges materials, usually sugars from the plant and minerals and nutrients from the mushroom. It's likely that other mutual benefits occur. There is evidence that mushrooms offer some protections and communication functions. Cortinarius and russulas are also mycorrhizal, but generally with different other partners. Okay, we can go to the next. This is Amanita flavoconia, called yellow patches. It's fairly common, actually. The image in the book shows a more mature group whose caps have expanded. Okay, we can go to the next. This is an unknown Amanita species with an inverted ring. The ring begins as tissue covering up the gills, protecting them, and then the cap expands and leaves the ring, so it tears away from the edge and leaves the ring around the stalk. The configuration that ring makes is an aid to identifying which mushroom you have, which Amanita you have. Amanitas start as an egg that expands, often leaving material, warts on the cap, as well as stuff at the bottom. That is a bulb, like you see here is a bulb, and sometimes a sac enclosing the stalk. Sometimes they're just ephemeral fragments at the base, so you really have to look carefully, and sometimes it's just buried, so you need a knife to dig it up. Okay. So mushrooms don't always grow singly. This Omphallotus illudens usually grows in big clusters on wood, and they can be really bright orange, and they glow in the dark too. So this is actually my daughter picked some, and she put it upside down because it's mostly the gills in her wastebasket, and used it as a nightlight. This is a species in the new book, but pictured with a different image. Omphallotus is poisonous. Next, Marasmiellus candidus is not featured in the new guide, but there are many small clustered as well as many small single mushrooms. And next, recent genetic evidence indicates that Pleurotus ostreotus, the oyster mushroom, may not occur in North America, but several similar things do. The white-capped Pleurotus pulmonarius, not shown here, has very pale purple spores and is pretty common in Kansas. But then this last December, I found a dark-capped Pleurotus, and its spores were pale purple. So they may be integrating. There's lots to learn about. Okay, so next. Cantharellus savarius is another species that probably isn't North American. It has its big distinguishing character is that it has ridges, not gills. So the gills in actual gilled mushrooms are separate structures. These are simply ridges from the cap. They can get pretty tall, so you need to look very carefully. In our woods, we find a variety of chanterelles, bright orange, golden, and almost white. They're usually small and low to the ground, but occasionally pretty large and all edible unless growing on something poisonous. And that's something you do need to think about. What is the mushroom growing on? Because they suck up poisons, herbicides, oil, lead. There were some people that died because they picked a very edible mushroom along an old superhighway. And there was lead all in there, and they ate the mushrooms, and they were perfectly the right kind of mushrooms, but they were thick with lead. So, okay, next. So this is Apiopyridon piriforme. It's one of the few puffballs that grow on wood, and it's my favorite edible puffball. And next, this is, gosh, I'm lost. Here we go. This is a small, lovely earth star, probably belonging to the Asteris genus because the petals are textured, and it's a rare mycorrhizal earth star that's not included in the book. Most earth stars, most puffballs, maybe all puffballs, are saprobes, and so they're growing on dead matter. Okay, next. And there are several hericium found in Kansas, and they all grow on wood and all are edible, again, with the caution that it's got to be pristine wood. Okay. And this is a bolete that has tubes with pore mouths on its underside. There are a bunch of red and yellow common boletes. Hortoboletus campestris is one of the smaller ones. It likes oaks. You can see them in town quite often. It bruises blue unless it's really old and dry. And you can see sort of the faint blue on the stalk, and that's probably from being picked. And next. And this is a different image of Boletus subvaluatipes than in the book. This bolete has red pores, though the tubes are yellow. Usually the red pores indicate a poisonous species, and that is the case here. And next. Polyporus radicatus has pores on its underside, but is tougher than a bolete. So it's not really a bolete, it's called a polypore. It has a black root below the ground that reaches down to buried wood. You can sort of see that. It's not really well illustrated. And next. This is a favorite edible if you aren't allergic to it. The white pores of Laetiporus cincinnatus distinguish it from the yellow ones of Laetiporus sulphureus, which is the one that's pictured in the new guide. Okay, and next. Termites versicolor grows on wood. It's a common thin shelf with white fine pores on its underside. You can kind of see that at the edge there. And next. Ansterium ostrea, the false turkey tail, has no pores at all. Instead of smooth on the underside, it still makes spores. Okay, and next. Tricaptan biforme looks a lot like the last two images we saw, except for the purple edge, which will fade with age, but the underside often looks tooth-like. So you need to look carefully at these the undersides of well, all of them, but these little shelves are mostly distinguished by what their fertile surface is on the underside. Okay, I'm going to have a drink of water. So we've got a lot of corals in Kansas, and some of them do make you sick, and some of them are pretty hot. I've never heard of anybody being killed by them, but again, you need to be careful. So next. This is Ramaria, a coral, Ramaria ariospora. I wouldn't recommend eating this, though some people have. So next. And this is Dacropinax spathularia, a much smaller than witch's butter, which maybe some people know about, but still the same sort of texture. It's jelly-like. It's like a glove of a plastic glove full of water, and it's just as yellow. And the next. And this is the first ascomycete. Lots of ascomycetes are cup-shaped. And while there are fewer basidios, many ascos are so small and obscure, I'm only going to show you a few here. Sarcosypha dudleyi is an early spring cup fungi, and the yellow form is extremely rare. I've seen it just once. And next. We used to think there were little grays and big yellows, but genetics tell us that they are the same thing. Just the grays are the young ones. Morels are always hollow in the center. So cut from top to bottom, they're going to be hollow. The false morels have stuffed stems or chambered stems. And next. And this is Dicina caroliniana is a false morel, and it may contain monomethylhydrazine, which is pretty poison. It's actually a rocket fuel. So pretty volatile. And some of them have this and some of them don't. And we don't really understand when they do and when they don't. Cooking removes much of the poison, but don't sniff the steam because people have died from doing so. Its stem is stuffed and chambered. And next. This is Helvela elastica. It's a non-edible cousin of the morel. And next, Helvela stevensii is much smaller than Helvela elastica. And occasionally I've seen them by the dozens in town in the spring. And this is a true tuber and found in Kansas. Tuber brennemanii it's a true edible truffle. And we have at least one more because I stole two from a squirrel. They turned out to be Tuber leonii, so not this one. Tuber are mycorrhizal, but there are a lot of underground fungi that are not true truffles, and some are poisonous. So that is the last of my slides. So if you've got lots of questions, that would be great.
Well, I know I saw some things in the chat. So EJ, I don't know if we have any questions. Otherwise, I'd say people could turn their cameras on and ask away.
Or you can ask without turning your camera on. It's okay.
You can ask without turning your camera on.
Thank you so much. That was such a good overview. Yeah, there were some questions that came through the chat that I'd be happy to relay. The first one was about fairy rings and what kind of mushrooms tend to grow in fairy rings, and that came from Jesse.
There are a lot of mushrooms that grow in fairy rings. And one of my favorite is Marasmius aureatus, which grows on Mount Oread too. It does. It grows lots of other places. And what happens is you have a point of infection or where they're growing, and they exhaust the minerals or whatever they need there, whether it's saprophytes are growing on dead grass, they exhaust what they need from that, and then they spread out, and that makes a ring. So there are a lot of them that do that, and I can't really name all of them that do. So, but Marasmius aureatus is my favorite, and it's an edible too.
Great. Another question from someone with HH as their name. Um, are there a lot of bioluminescent mushrooms in Kansas?
Are there a lot? Well, there's sure some, and I saw some. It's a little tiny one, and I'm trying to remember what it's called. Gosh, it's Stypticus. It's well, that's its specific. Sorry, I'm blanking on it. And it's small and shell shaped, and they need to be moist to luminesce.
It's
And of
Pinellas.
course, what?
Pinellas.
Well, Pinellas, thank you. Thank you.
Caleb that's on.
Oh, Caleb's on there. Oh, good for you. Okay, Pinellas. And I saw some of that. The mycelium of the armillaria are luminescent. And I think it's one of the things that's called foxfire. I think there are several things that are called foxfire. But I know a friend was driving in the dark and saw something that he thought was fire, and he went over, and it was a dead tree uprooted, and it was just a maze of this greenish bioluminescence. So let's see. Well, the Omphalotus, is it Olerius? I can't remember. Caleb?
Can I just weigh in about the fairy rings?
Sure, sure.
The most common thing I see forming fairy rings in Lawrence is chlorophyllum
Oh, yes.
studies. And it
Yes.
forms magnificent fairy rings in lawns.
Mhm.
There's one near my kid's school that starts on one corner of the intersection and extends, I think, under the street and shows up on the other corner of the intersection.
Wow.
And that I think Sherry said was the most common poisoner in Kansas. They look delicious, but they're not. But they do make really beautiful fairy rings.
Thank you,
Yeah,
Caleb.
on that too, there was another question from Jesse. I don't know which species they were referring to here, but I'm curious about if pores indicate poisonous mushrooms or if there anything in particular that might indicate that mushroom might be poisonous.
There is simply no one thing, just not. There's just no way you can do that. You really need to look at the whole, and yeah, it's a whole kingdom. So you're talking about variety of the sort in the animal kingdom of blowfish to giraffes. I
Mm.
mean, how do you? Giraffes don't have scales, you know. So there is simply not. And I know that there was the silver spoon idea. There are a lot of, but you just cannot go by that any one thing.
Great information. I have heard in the past one rule of thumb was that if a mushroom is growing on a tree, it is more likely to be safe than a mushroom growing on the soil. What would you say to that?
No.
Great.
That's not true. There's a very Galerina marginalis is a charming little mushroom with a beautiful tailored ring on it, and it will kill you dead. I mean, if you eat it, you can touch it. There is no mushroom that's going to hurt you by touching it. And in fact, most gosh, I've tasted, I think, a lot of mushrooms and spit it out, but sometimes the taste tells you something about it. Don't keep it in your mouth, don't swallow. And in fact, that's one of the things I should say is if you're trying a mushroom and you're really certain it's an edible, you might have a sensitivity to it. Allergy or who knows what. So if you're sure it's the right kind, I would saute just a little bit of it, eat a little bit of it, and wait 48 hours and see if you've had some tiny adverse reaction. So anyway, I had a good friend who really wanted to try mushrooms, and I cooked up a bunch of latiparus as hors d'oeuvre for dinner. And I said, I want you to eat one piece of this. And she did not. And she swelled up bright red, and she couldn't go to work the next day, but she was okay. So you just never know. And I had somebody who ate chanterelles, which I just can't believe is going to be a problem for anybody, but her lips got numb. It was disconcerting to her. She wasn't really harmed, but nevertheless.
All very good to know. I guess building off of that too, Matt is asking, what is the most common slip up, misidentifying common edible mushrooms in Kansas?
The most slip up. Well, since there's so many there's so many ways to slip up. You just need to be careful about what you get. I actually personally have picked an Omphallotus alludens that was straggling away from the group. I picked it up as a chanterelle. I myself, I didn't eat it. After I picked it up, I looked at the underside, and it had these blade-like gills instead of the ridges that the chanterelle. There were chanterelles all around, and they were just the same color. But so that's the only thing I can think of. I think people will pick false morels thinking they're maybe not the same, but are good to eat. A lot of people do. There are several false morels, and I just wouldn't recommend it. I actually have eaten them. They're good. It's not worth it. They're in grocery stores.
I think in the book, Sherry, I think in the mushroom preparation section, Sherry makes a really good point that even if you're a pretty good forager and you know what you're looking at, it's easy to find mixed populations of different species that kind of look the same, and it's a really good idea to examine each and every mushroom very carefully to make sure that you don't have a mixed population of an edible and an inedible species before you start cooking them.
True.
Okay, another question from Matt. He was wondering if you said that cedars are the tree you most associate with morels.
No. Morels are what you call promiscuous, and they like a lot of trees. In fact, I was surprised to find them with cedar, but this was just an incredible crop. I mean, there must have been, really 100 pounds of morels in this area, and I've rarely seen them with cedar since. I think it's cottonwood, apples, spruce. I've been told they like spruce. They're where you find them. They're flexible. What I think probably happens, well, it's a little debatable, but they have a divided lifestyle. So some of the time they're mycorrhizal and they have a host and they're doing just fine. And then something happens to the host. It's injured or something. At that point, they think they've got to get out of there. So they put up their fruiting bodies at that point. And then also the organism can break down the dead wood. So there are a little of each. And we're discovering it's not one or the other. Like the rest of the world, it's just not black and white. So things are not only mycorrhizal. Sometimes they're mycorrhizal for a while, and then they become saprophytes, or maybe even parasites. Now we're using a term called biotropic, which means living on, yeah, well, anyway, it's more general. So.
Fungi are so cool. There's never one answer, it seems like, is what I'm learning.
No, that's a real cop-out, isn't it? I can say whatever. One of the things I would like to say is that a very good way of one of the ways of identifying mushrooms is to get a spore print. And I did not tell people how to get a spore print, and it's quite easy, though sometimes the mushroom does not cooperate. Basically, you want to put the fertile surface down on a piece. I like white paper, but if you have something with a ISBN number, so it has black bars across it, because you get white spore prints and so you put the fertile surface down on that. You put a damp cotton or whatever on top of that, and then a little container over that. And some things drop their spores in a couple of hours, and sometimes it's overnight, and occasionally not at all. And it's just mysterious. Don't refrigerate your specimen before you try to get a spore print, because that really does discourage them. I know Russells almost never will drop their spores after they've been in the refrigerator.
Great. Okay, more good questions from HH. Asking if every bright colored mushroom is poisonous, and then also wondering if mushrooms mimic others, like non-poisonous mushroom posing as a poisonous one.
Well, I'm not sure intentionally, but what was the first question? I'm sorry, I didn't quite understand that.
If every bright colored mushroom is poisonous.
No, the chanterelles are a good case in point. And then there's an incredibly beautiful blue milk mushroom. It's a Lactarius or Lactifluus. And I mean, it's brilliant blue. And you break it and it exudes this thick blue milk, which turns green. And it's a good edible. So a lot of red ones, but that you just know one rule. There was a second part, and I don't think I.
About mushrooms mimicking others, like a non-poisonous mushroom posing as a poisonous one.
Well, I don't think they're posing as they're not hoping you eat them and die and propagate or something. No, I mean, I think of monarchs and viceroys. That's intentional. I mean, as far as evolution goes, but I don't think mushrooms are doing that. But you still can mistake poisonous for non-poisonous.
And it's not the case that mushrooms that are toxic to humans are universally toxic. There are many animals that devour them without apparently any negative effects.
Or they go off and die.
Or they go off and die.
I mean, you don't know. Yeah, they have bites out of them, and you don't. And there are actually some. Well, I'm not thinking of mammals, but I think there are some that are immune to some poisons. But certainly there are some Amanita flies that have maggots that particularly like Amanitas, and I think they probably are picking up the poisons, just like the monarch is picking up the poisons from the milkweed.
I think that covers most of the questions in the chat, but I have a question for you. I spend a lot of time in the woods, and it sounds like you do too. And
Yes.
I'm always just, learning while I'm out there, seeing things that interest me and, seeing some cool relationship between plants and animals. And so I'm curious if there's anything that you have learned just from being out in the woods that has really surprised you.
Oh my goodness, I could write another book on that. I mean, anything particularly? Oh, we went to the goat ranch. I think that walk. I inventoried that area about 10 years ago, and I went out by myself, and I was finding lots of interesting things, and I noticed some, I don't know, movement. And there was a little doe, and she was watching me. And I thought, well, fine. She watched me, she followed me for maybe 2 hours. I don't know what it was about. She was charming. But that was a real surprise. I mean, that was a real, I mean, there are so many things that happen everywhere. But I think in nature, maybe particularly, because, I mean, we're used to being in our little cocoon of human community. And so yeah. So, I mean, that's
a great question, EJ.
Yeah, thank you. That's wild. I wonder what that deer saw in you.
I just have no idea. I wasn't, I don't think I was, well, I don't think I was threatening in any way, but, I was probably behaving strangely because I'd bend over and do something and then I'd go on. I don't know.
You mean not every person is as excited about mushrooms as you are? You don't say.
That's maybe the doe didn't think so. Though I think deer do eat some mushrooms.
Another question I have, I know folks getting into foraging, it can be very overwhelming for plants on one hand, that can be intimidating, but then mushrooms kind of feels like a next level because there are some that can be really toxic. So what is your advice to somebody who hasn't done much foraging in the past and is looking to get into it, or maybe has done a lot of plant foraging but is looking to start trying some mushrooms?
Well, I think it's best to go out with somebody who knows or bring the mushroom to somebody who knows. And actually, tomorrow, the Kaw Valley Mycological Society is going to go out on a foray. We're going to meet at, well, it's a Wyandotte, is it County Lake, I think, in Wyandotte County. And it's a big park, but we're going to the north side where the spillway, it's a spillway parking lot. We're meeting there at 10 o'clock. And really, anyone is welcome to come along and do that. Mushrooms have their seasons. Actually, there are not as many different mushrooms or quantity of mushrooms in the spring. And then there's sort of a hiatus in late May and in June. And then the summer mushrooms come on, and then you have much, much more variety and quantity too. And that goes into the fall. So if we get rain in the summer, that's the best time. So I don't know what we're going to find tomorrow. I can't guarantee morels. I will probably see at least some old wood rotters and stuff. But anybody who wants to come along, it's fine. We don't even take dues anymore. So just show up. And I mean, we will leave at 10 though. So
that sort of feeds into a question I had about in the book when you're talking about Kansas mushrooms specifically. You describe it as feast or famine.
Yes.
And so what are you looking for? Is it a date on the calendar? Is it a specific kind of weather when you think, oh, there's no way I could go to the grocery store today. I have to go look for mushrooms. What would that day look like?
First of all, I should say I'm not much of a forager anymore. I've had them rotting on my table too often, so I don't. But so I probably don't ever quite say that, though I really am excited about going out and finding things and finding new things. But as I say, there's a hiatus in May and June, and if I get rains around the 4th of July, I am going to be out in the woods. It'll take several days. I wouldn't go on the day it rained unless there'd been some rains earlier. And I think things look pretty good since we had this nice big rain, and then we had a little rain before that. Now it's going to get really cold again, but some mushrooms are encouraged to fruit at that point. I think it might be a strategy because you don't have as many bugs in.
Right.
But and it depends on what you're looking for. I'm not going to be looking for morels in the middle of the summer. In the fall, you find something called Grifola frondosa, hen of the woods. And you're not going to find that in the summer. In the summer, you're more likely to find the latiparus, these bright orange with yellow on the underside, or muted orange with white on the underside. So, mushrooms are where you find them, when you find them. And, the weather's been strange, so it's sort of hit or miss. You don't know that it rained up where you're going to hunt, which is 3 miles away, even though it rained in town. So it's hard to go out when you're having fun, when you feel like it. Take a basket, whatever. Prepare for ticks. I really recommend spraying your clothes with pyrethrin, which you can usually get, well, I think you can get it at Lawrence Farm and Feed. I'm not sure where else. But some of the other sprays really don't do as well. Actually, I just sprayed this because I'm going to go out tomorrow.
So you're ready. There's any ticks in here?
No ticks. Well, my dogs bring them in, actually. So, and they're immune, but not me.
That's great. Well, does anybody else in the group have a question that they would like to ask Sherry? Or a comment? Ryan Armbruster put up a delicious picture of morels he found, so I'm surprised he hasn't unmuted to brag about that yet, but
see, I can't.
I will unmute just to say we ate the last of the basket of morels this afternoon for lunch, and it was bittersweet because there's something nice about having a few morels in your fridge, and now they're gone. So
like the money in the bank.
I did have a question, I guess, as long as I'm unmuted. I've got a buddy who asked me this last week. He's actually the same guy who gave me the morels. He was asking if there's anything that can be done in terms of management or trying to plant some seedlings or anything, so he can even sweeten his honey hole of morels so he can ensure that even more are out there in the future. And I had no idea what to tell him, so I figured I'd ask you. And most morels are found, on at least around here on public land. So I'm not sure how much management you can really do. But if someone wanted to ensure that if they had morels already on their land, if they wanted to get more, are you aware of anything that you can do to really get more out there?
Well, the problem is that I think that morels usually fruit when the trees are dying. And I've never seen a morel around a dead tree that didn't have its bark on, so newly dead or injured trees. But I hate to recommend, especially with the Forest Service, when you take an axe to your trees. I don't know beyond that. There have been several attempts to grow morels. Some of them supposedly fairly successful. You could check with Paul Stamets. He has a website called Fungi Perfecti and see what he has to say about it. But they're pretty elusive. They're where you find them, and that's about it. Sorry.
You guys want to hear a funny morel story?
Yes, Stephen, please.
So last fall, when my brother was here visiting from Denver, I took him out to Kaw River State Park to show him where we'd been working on doing the habitat restoration, to show him all the bush honeysuckle that we'd removed. And so we're out there. There was this guy out there. He was gathering grapevines that he was using to make wreaths out of, which, he probably shouldn't have actually been doing that. But anyway, so I'm telling my brother about, it's like, look how, much we've improved this area out here. And the guy heard us talking and he's like going, oh, he goes, oh, you're the people that have been doing that? I'm like, yeah. And he's like, you guys are ruin. He's like, I don't know what you're doing out here. You're ruining it for other people. And I'm like, we are. He goes, you guys ruined my morel hunting spot down here by cutting down all of that brush. And, uh, and I said, well, he's like, I'm like, really? He goes, yeah, I went down there this year and there weren't any morels there at all. And so, I had to explain to the guy, it's like actually removing all that honeysuckle is going to improve the habitat for your morels, right? But, I didn't have the heart to tell him that, now that jungle of honeysuckle is gone, people can come out here to the park and it's pretty easy to see them down there now. Well hidden before, but hey, I'll just let him deal with that. But anyway, it cracked me up. He's like, yeah, you guys are just ruining it out here. And I'm like, I'm like, it may seem like it, but the two really aren't, directly related.
There is a plant called, what is it? Mustard. What is it, Caleb?
Oh, garlic mustard.
Garlic mustard, which is said to interfere with mycorrhizal associations.
Yeah, well, there's a bunch of that out there too, but we've been ripping that up too.
So. Good for you.
Yeah,
I do it every
another
chance.
invasive.
Absolutely. Yes, yes.
Another question came in from Jesse, and they're asking, how do you know if you're allowed to forage mushrooms or if the soil is safe from chemicals?
Ask. And who to do that, I don't know. I mean, if you're on, I mean, you could talk to the forestry people. I mean, you guys don't allow spraying in parks, do you?
That's not anything that we have any jurisdiction over. We don't own any land.
Okay.
I will say if it's your land, you could have your soil tested for lead because you mentioned that, and your local extension office will do that. But private land that isn't yours, I think, is a major challenge. And we talked about that in the book club EJ did on Last Wild Places in Kansas, how challenging it is with the very, very limited public lands that we have.
Yeah. Yeah, test it. Ask, what the land's been used for, and that's don't pick along highways. I mean, that's just I've had people say also picking garlic in ditches. I have a feeling that's not a mushroom, but I have a feeling you probably shouldn't be doing that. Mm-hmm. I mean, that's if it's an old road.
One thing on the safety note that you mentioned in the book that I thought was so interesting is that you shouldn't go out and pick a bunch of mushrooms and then bring them home and try to identify them. But that when you're out, you should actually identify them where you find them. Why is that?
You should try to.
Right.
Or make notes. And I tend to not do that because I want to, but the safest thing to do is to make notes about, because they change color. I picked this bright purple mushroom. Well, this was on a national foray, and it was large and it just beautiful. I put it in my basket, and really in minutes it was gray. Okay. So they change color, and you really need to isolate your specimens, because if they bat about against each other, they will color or change each other. So I recommend a basket and wax paper bags, not plastic bags. Plastic bags, you put mushrooms in them, and they're more likely to mature or rot because it's an enclosed, warm area. Now, in the spring, you often get away with that. I think morel pickers do, but I just wouldn't recommend that. So wax paper bags. So isolate your specimens. Look at them when you first get them. A lot of mushrooms stain. So you don't want to look at them when you get them home too, because they will change. So a lot of mushrooms stain. So look at them right away. Dig them up with a knife or a trowel if you really feel like you need to see what other structures are underground, especially with Amanita, but other things too, like that Polyporus radicatus, which actually is an okay edible when it's really young. But you just don't know what it is until you dig it up. So observation in the field is the very first start to identification. But you don't do it all together then. You bring them home and take a look at those spore prints. I mean, you're not going to, actually, if I'm traveling a distance, I'll have things set up for spore prints so that I start them. And by the time I get home, sometimes I have spore prints if it's 2 or 3 hours away. Yeah, in field observations are really important.
Yeah, the other cool thing with mushrooms too is that you often have to use so many more of your senses. Like obviously you have to use your eyes, but yeah, touch, smell, taste, if it makes sense, all of those things. It really widens your horizons in terms of plant and mushroom identification.
Yeah, there are many dimensions. Yeah, I wouldn't for sure identify anything just by an image. I could say it looks like this. You go from there.
Yeah, along those lines, some artificial intelligence programs are really good for identifying plants and birds. My experience is that they're pretty lousy still for identifying fungi of all kinds.
Yes, I
in
agree.
large part, I think, because what EJ said is that they require so many other characters that are not just visual in order to correctly identify them. So you can't rely on iNaturalist to identify a mushroom.
That's true.
Well, and that's a really good point because I think, a lot of people, if you want to use that to identify a weed in your lawn, what's the real risk there if you get it wrong versus something you're consuming? That's a great point, Kayla.
Right.
Another reason to buy the book and not just use the app.
Yeah.
That's right. This is all about that.
Well, and I think that's a good note to end on because we are right at an hour. So if you haven't already, pick up A New Guide to Kansas Mushrooms. It is, even though, nice, EJ, I love that. We got it right on the screen. So even though a couple of us on the call are K-State employees, this is a University of Kansas Press printed book, and we're very lucky to have that here in the state. So we'll support KU with this book, which is excellent. If you haven't gotten it already, please do so. Thank you, Caleb, for being on.
Thanks,
Thank
Caleb.
you, EJ, for moderating today. Thank you for everybody that joined, and thank you so much, Sherry.
You're very welcome.
For being on.
It was fun.
It was a wonderful chat. We hope you all join next time and let us know if you have any questions. Thanks, guys.
Bye.